Food preservation. Food preservation can be defined as a process of treating and handling food in such a way as to stop or greatly slow down the spoilage and prevent the foodborne illness while maintaining nutritional value, texture and flavors. So when we are doing the preservation, we have to keep in mind that we need to maintain all the nutritional value, texture and its flavor. Retaining food over a long period of time without being contaminated by pathogenic organism or chemicals and without losing its. So this is the definition of the food preservation. Now why the food preservation is required? Because of the seasonal availability of the food. Some foods are only available during their seasons, favorable season. So if we want to consume it afterwards also, if we want to need it, require it afterwards also, we need to preserve those things. And different type of food at the different locations. So by preserving food, we can carry out the food from one location to other location. Most food deteriorate in the quality following harvest, slaughter or manufacture. So this way the conditions should be properly kept so that the preservation of the food can be done. The principal quality deterioration reaction of the food may be. So here we will see what are the different type of the deterioration. So first is the microbiological, that is due to the microorganism, and that is caused around 10 to 25% in the various type of the food by the microbial losses. So second is the enzymatic by its own enzyme. Okay, so enzyme native to the plants or animal tissue. So they will be responsible for their spoilage, deterioration. Then chemicals. So chemical reactions like oxidation, rancidity, oxidative reduction, reductive discoloration, non-enzymatic browning, destruction of the nutrients. So all these are the chemical deterioration. Then the physical, that is loss of the texture, flavors and structural damage. Now, what are the advantage of the food preservation? So let's see, that is the increasing of the shelf life, retaining the quality of the food, increasing the food supply, decreasing the wastage of the food. So all these will be the advantage of the food preservation. Now coming to the principle of the food preservation. So what are there are three different type of the principle. Firstly, by preventing or delay of the microbial decomposition. So this is done by the asepsis, removal of organism, hindering the growth and activity. Second is the prevention or delay of the self-destruction or the self-decomposition of the food. So that can be done by inactivation of the food enzyme. That is, because of food own enzymes, they will self-decompose. So if we improve those factors, like if we inactivate those food enzymes, that can be slower down. Okay, so third principle is the prevention of damage because of insect, animal or the mechanical causes. Now coming to the different method of the food preservation. These are the different methods of the food preservation that we will see one by one. First is the asepsis. So as the name indicates, asepsis is the keeping out microorganism as a preservative factor. So we will keep asepsis, we will keep the microbes away. So bioburden of the microorganism. Okay, so that is the bioburden, that is the normal microbes present on any of the food item. Okay, and now depending on the food, the kinds of the organisms are also variable. Now number of the microorganism also very important, because more the spoilage, more likely the organism are there. Okay, so if the food is having more number of microorganism, there are more chances of the food spoilage easily. So bioburden may be the result of contamination, growth of organism or both. Now the quality of the many kind of the food is judged partly by the number of microorganism present. So this is the first method in which we are looking for the asepsis, how we can keep the microbe away. So asepsis methods are directed towards avoiding and minimizing the microbial contamination. So how we will do it? Firstly, packaging food. So that is done by the aseptic method. So here what we are doing, we are preventing the contamination during handling. So what we are doing, we are trying to keep the microbe away. Then in the dairy industry, here also we are doing the proper hygienic practices. Third is the canning industry. So here also we are doing under the sealing, under the proper heat conditions. Okay. Then next is the meat packaging industry. Here also we are doing under the proper hygienic condition, proper sanitary methods. Now other industries also control also have the controlled fermentation, like cheese industries. All these are also here we are keeping the asepsis practices. Like here if you see the cheese industry, if we are not doing the controlled fermentation, that fermentation which is required for the production of the food will result in the contamination of that same food. So for that also we are keeping that preservation is also there, that is the asepsis. So we are doing all these things under controlled organism, under controlled condition, under controlled fermentation. So this was the first method of the preservation. Secondly, if you see the removal of microorganism, so removal is also accomplished by the filtration, centrifugation and washing. So as the name indicates, by filtration also we will do, then by the centrifugation also we can do, and then the washing can also be done. Like when we are getting our things, we have to firstly wash them, and fourth is the trimming. So that is the spoiled portion of the food can be discarded by the trimming. So this removal of microorganism, that is helpful in the partial. It is not going to completely remove the microbes, but it can preserve the food for some time. Now next method for the modification of the organism, of the environment, organism environment. So modified atmosphere can be defined as one of the specially created. So when we are altering the natural environment, natural distribution or the makeup of the atmospheric gases where the microbe is present, if we modify it, then that is called the modification. So we are altering that environment. How we are doing it? We can do it by vacuum packing. We can do it by modified atmosphere packing. We can do by controlled atmosphere. So these are the three different procedures. Now, firstly, the vacuum packing. So what we are doing here we are evacuated the bags in which we are sealing or in which we are packaging the food item. So what it will do, the residual oxygen that is packed inside with the food, that will be absorbed through the chemical reaction and the residual respiratory activity in the product and its microflora. So that is used for the very primary, like meat or fish, prepared salads. So all these things, we all have seen these type of packages. So the activity of the microbes, because the residual oxygen will be absorbed and very less activity will be there. So that is also one of the environment modification. So here vacuum packing accomplished three things. First is the displacement of oxygen to delay the oxidation. Second is the decreasing the growth of aerobic spoilage organism. Third is the acting as a filler to maintain package conformity. So that is there. Next is the MAP, that is modified. So the bulk retail package, it is flushed through with a gas, usually mixed, usually containing some combination of carbon dioxide. So here what we are doing, we are adding carbon dioxide, oxygen and nitrogen combination inside it. So by this the oxidation will be delayed. Okay. Then is the CAP. So here also we are doing we are maintaining the environment, we are controlling the environment through by storage. Okay. So we are providing it during the bulk storage or the transport of the food. We are using this controlled atmosphere. Okay. So these are the three methods for the modification of the environment. Fourth is the use of high temperature. So fourth method which we are using for the preservation is the high temperature. High temperature is very important. Use of high temperature. So what is the principle behind it? So what the high temperature is doing, it is destructing the effect of the microorganism. Okay, so microorganism when they are destructing, when their destruction is there, so what it will do, it will kill down, it will die down, so the number of the microbial load, the amount of the microbial load, that will be reduced and the preservation can easily be done. So here in this graph also we can see the number of cell and the increasing resistance with the increasing temperature, how it is working. Now the temperature and time used in the heat processing of food will also depend on what effect heat has on the food and what other preservative methods are to be employed. Yes, so when we are using the heat, that is also very much, if other methods are also there, then that also will vary. Okay, so if not all organisms are killed, either the heating must be destroy all potential spoilage microorganism, or the food must be handled thereafter so as to delay or prevent the growth of the surviving spoilage organism. So this way the heat treatment is very important and it will sterilize or it can preserve the food for very longer duration. So there are now, for better understanding, we will see that how we are using this heat for the preservation. Firstly in the pasteurization, second heat at about 100 degree, third heat above 100 degree. So firstly we all know about the pasteurization. Pasteurization means what we are doing, we are killing the organism, not all. Only pathogenic are going to be killed, and it is also below 100 degree. Okay, so heating may be by means of steam, hot water, dry heat, electric current, and the product are cooled promptly after the heat treatment. Okay, so when the pasteurization is required, when more rigorous heat treatment might harm the quality of the product, when aim is to kill the pathogen, when the main spoilage organism are not very heat resistant, such as yeast in the fruit juices. So all these are the different purposes why we are using the pasteurization for some food. Now preservative methods used to supplement pasteurization. So what are the different foods that we are using? These are the different foods in which we are keeping them, preserving them by the pasteurization method. Now, the time and the temperature used in the pasteurization also depend on the method employed on the product treated. So, what we are doing, there are three types. Firstly, it is the high temperature short time. So, that is 71.5 degree for 15 seconds. Second is the low temperature long time, that is LHT, that is 62.8 degree centigrade for 30 minutes. And third is the ultra high temperature, when it is 138 degree centigrade, and this is for 2 seconds. Okay, now heating at about 100 degree. So when we are doing it at about 100 degree, so it is almost 100 degrees obtained at that temperature. So this can be done, but it is not more than 100. Okay, it will be below, in between 60 to 80 to 190, so like this only. So in this you can use, you can see the baking, simmering, roasting, frying and warming up. All these things are done for the food preservation. Now heating above 100 degree. So above 100 degree, this is by the, generally it is done by the steam, and that is also under pressure. So that also will be done. So these are the different temperature and the pressure when we are doing it. Okay, so this is the high temperature used for the food preservation. Now coming to the low temperature used for the food preservation. Low temperature are used to retard the like high temperature will destruct, but low temperature, what they are doing, they are retarding the chemical reaction and the action of the food enzyme, and they will so in resulting in the slowing down or the stop of the growth and the activity of the organism in the food. So when the temperature is low, slower the growth of slower the rate of reaction, slower the enzyme action, slower the microbial growth, and it will prevent the growth of organism, and the proper preservation will be there. So here what will happen, like cooler temperature will always prevent the growth, and but what will happen, activity will continue, slow, very slow metabolic activity will continue. So the cooling of the food from ordinary temperature has different effect on the various organism present. Now what will happen, a drop of 10 degree may stop the growth of some organism and slow the growth of other. Now further, if you decrease the temperature 10 degree, that would stop the growth of some organism and still slower the growth of so like this you can understand that different temperature will have the different effect, that is depending on the type of the organism present in the food. Now what are the different, how we are using the low temperature? So we can be using it at the like common storage, chilling or the cold storage, and third is the freezing or frozen storage. So all these three are the category for the use of the low temperature for the food preservation. Firstly, the common storage. So here what it is happening, here it is generally like we have heard for the cold storage, when we are storing the things less than 15 degree, like fruits, vegetables, these things. Okay, then cold storage. So cold storage is also like when we are keeping it in our fridge, refrigerator. So that is typically below 8 degree. So here what is happening, here also we are using the perishable foods and dairy products, all these things. Then freezing. Freezing is technically, that is when the temperature is less than 0, and generally it is minus 18 degree or minus 20 degree, and this will be done for the food items which are kept for the long term preservation. Okay, so this was the use of low temperature for the food preservation. Next, coming to the drying or the water removal. It is also one of the very important one of the very important method for the food preservation, because drying, Drying, what it will do? Drying will remove the water, and when the water is removed, when the moisture is removed in the food, the activity of the microbes, activity of bacteria, yeast, mold, that will always be inhibited. Okay, so this can be done by sun drying, electric drying, and freeze drying. So during here also you can see that sun drying, this is the electric drying, this is the freeze drying. So smoking also, smoking drying, drying during smoking is also one of the important type or the method for the drying. Okay. Now the decreased water availability. So by the, when it is also one of the, like drying we are doing with the, in that method, when we are sun drying it, when we are machine drying it, and when we are frost drying it. So there also we are inhibiting the activity of microbes by reducing the water content or by taking the water content out of from the food. Same way the water availability will decrease by three other methods here also: dehydration, crystallization, and addition of the solute. So these are the three methods by which the water availability will be decreased. So the water activity is used for the preservation of the food, stabilization of food supply, and developing different type of shelf stable foods. So these are the different methods. So reduction of the water, what it will do, it will prevent the growth of the vegetative microbial cell, germination of the spore, and toxin production. So this is the principle by which the decreased water activity will inhibit the growth of microbes. So decrease in water activity, that will increase the lag phase of microbes and decrease the growth rate. Okay, so this is the decreased water activity method. Coming to next and very important is the chemical preservative. So different type of food additives will be added, that will be to preserve the food, and that will prevent the deterioration, decomposition of food items, and these are known as the chemical preservatives. So what they will do, they will the deterioration of food that is caused by the microbes, it can be stopped or it can be prevented by the use of these preservatives. So food additives or a preservative is a substance or a mixture of substance other than the basic foodstuff which is present in a food as a result of various aspects of production, processing, storage, and packaging. So during the production, during the processing, during the storage, these preservatives will be added. Okay. So the inhibition of the growth and the activity of microbes is one of the main purposes of the growth of the main purpose of the use of the chemical preservative. So what they will do, they will inhibit the microbes by interfering their cell membrane, their enzymatic activity, and their genetic mechanism. So these are the three things what these preservatives are doing. They will interfere the cell membrane, they will interfere the enzymatic activity of the microbes and their genetic mechanism also, by which they will inhibit the microbial growth. So these are the different factors for the effectiveness of the different chemical preservatives. Chemical, kind and the number of number, age and the previous history of organism, temperature, time, all these things. Okay. Now coming to what are the different preservatives can be used. So like salting. Salting, we all know that we are adding the NaCl or the sodium chloride or the common salt, and it will be, what it will do, it will draw the water out of the bacterial cell and the cell will be dehydrated, and then it can preserve. Okay, so these are the example of the food when we are doing it. Sugaring also, here also it is the addition of the sugar, that is also very important for, and it is also similar to here like the salt, it is also doing the removal of the water from the food. So for this, at least 65% of sugar solution is required. Next is smoking. Smoking is also one of the oldest method to improve the quality of the food, and it is commonly preserve the meat and fish. So what it is doing, it is also combined method like salting and drying can also be done with the smoking. Then spices also can be used. What like spices such as sage, they will exhibit the antioxidant and antimicrobial property. So such spices, if are added with the food, they will extend or they will prolong the oxidation process of the food, and such the spoilage will be prevented. Then the acids. So acids, different acid like citric acid, acetic acid, and ascorbic acid, they will also protect the food against the deterioration. Okay. Then acetic acid. Acetic acid is also very important, this preservative. And then other acidulants like malic acid, tartaric acid, citric acid, all these acids are also used for the food preservation. And then benzoic acid. Benzoic acid, it is very important, like it is used in the form of its sodium salt, and it is used in syrups, jam, jellies, sauerkraut, pickle, preservative, fruit cocktail, and different. So benzoic acid will be used here. Then sorbic acid, it is used in the form of sorbates, and these are also in the jam, jellies also, they are very much used. Now potassium sorbate, and all these are the used for the preservation. Now gaseous chemical food preservatives. So in this form, gaseous chemical food preservatives mein, here we can see that sulfur dioxide or sulfides, carbon dioxide and chlorine, all these are used for the as a gaseous chemical food preservatives. So these are the different type of foods, and these are the different conditions, different pH, how they are used, and they are different concentration also that will affect the food for its preservation. Then alcohol is also used. Then antibiotics also used when added with the food, like in the poultry, meats, fish, all these, they will lengthen the storage time at the chilling temperature. Okay, so here you can see so many combining preservative methods are used. Like we are not using one method, we can combine the different measures to get the proper effect or the proper result. Then one of the very important is the irradiation. So it is by the use of the radiations when we are preserving the food. So here the entire spectrum of radiation grouped into the two categories. So low frequency, low wavelength radiation ranges from radio waves to infrared, and the effect of these radiation of the microbes is related to their thermal agitation of the food. Same way, the high frequency and the shorter wavelength radiation, they will have high quantum energies, and actually they will excite or destroy the organic compound in the microorganism without heating the product. So the microbial destruction without generation of heat, without generation of high temperature, that is called as the cold sterilization. That is very important fact. So ionizing bactericidal efficacy of the given dose of irradiation, that will depend on the kind of the species present, number of the microbes present, and the composition of the food, presence of the oxygen, physical state of the food, and the condition of the organism. So irradiation can kill bacteria, mold, insects, pests, and it can reduce the ripening time, and this spoiling of the fruits, and at the high doses induce sterility. That is also very important. At high doses it can induce the sterility. So the proper dose should be given. So these are the some of the changes like that is brought about by the radiation process. So here you can see in the meat, the rise in pH, destruction of all these things will happen. Fat may and lipids may destruction of natural antioxidants, in the vitamins also. So all these things will happen during the irradiation sterilization. Okay. So if you see how the irradiation will affect, so directly when ionizing radiation will kill the microbe by damaging the biomolecule of their cell, then the incoming photon, it will hit the electron in the atom of the microbes or the food molecule, that biomolecule. Then during the collision, the photon's energy is transferred to the electron, changing the photon's direction. So here what will happen, electron free to collide with the neighboring electron. So collision will take place. So they will collide with each other, and it will cause the chemical bond breakage, and it will interrupt the normal cell metabolism and the division. So when the bond of the biomolecule will break, it will interrupt the normal cell metabolism, and it will result in the destruction of the microbial cell, and that way it will kill the microbe, and this way it can preserve the food for microbial contamination. So these are the different ways how the food can be preserved. So these are the different methods for the food preservation techniques. Thank you.